will wait 1 or 2 more minutes and we'll be starting soon. Okay, maybe let's start while people are joining. Welcome everyone, and thank you as every month for joining the joint seminars of cardiovascular research and the ESC Council for Basic Cardiovascular Sciences that we co-organize, and welcome and thank you Christian Weber, the chair of the council, who is co-chairing this webinar with me today. Today's topic of the webinar was chosen to be cardiac remodeling, cardiovascular remodeling in general. And we have two excellent speakers who have contributed enormously to the field: Eva van Rooij, who is at the same time deputy editor of Cardiovascular Research, and Sebastian Cremer, who will present a second presentation. Let me introduce the first speaker, and probably everyone knows Eva. She's a group leader at the Humboldt Institute and professor of molecular cardiology in Utrecht. She has already contributed hugely to understanding of various molecular signaling pathways that regulate cardiac and cardiovascular disease using mouse models, models of heart disease, novel sequencing technologies, which allowed her to identify key cell types and various molecules regulating cardiovascular pathophysiology. She has worked in a number of leading centers for cardiovascular disease, including Erik Olsson's lab, where she completed postdoctoral fellowship. Welcome, Eva, and thank you very much for working with the journal and for joining us here today. And please, if you can share with us some new exciting data, we're looking forward. Thank you.
Thank you very much, Tom, for that kind introduction. And indeed, what I'll do today is share with you some of the technologies we have been using to identify new mechanisms that we think might be relevant for cardiac remodeling. So one of the disease indications, cardiac disease indications we are interested in our lab is genetic heart disease. So of course, there's different type of genetic heart disease caused by different mutations in different set of genes. And these differences in phenotypes are really intriguing to us. And as they are so different, there's probably different molecular mechanisms underlying these remodeling responses. And we think it's an exceptionally exciting time based on all the technologies we now have available to us to identify new mechanisms that are actually relevant for disease progression. So one of the diseases we are interested in our lab is arrhythmogenic cardiomyopathy. It's also called a disease of the desmosome. So desmosomes are basically forming junctions between your cardiomyocytes. So they're really causing the integrity of your heart muscle. So normally a desmosomal complex comprises of several proteins like plakophilin, desmoplekin, and plakoglobin. And patients that have ACM often have an underlying genetic cause, mainly a mutation in one of these desmosomal proteins. So what happens in response to these mutations is that your cardiomyocytes detach, and then there's also an ongoing loss of cardiomyocytes, and these cardiomyocytes are being replaced by fibrofatty patches, and these patients also suffer from arrhythmia and sudden cardiac death. But actually how this all comes about is very unknown still. So we undertook an effort in our lab to try to understand these remodeling responses better. To do so, Kees-Jan Bogert, a postdoc in our lab, made use of the explanted ACM hearts that we have available at our biobank at the Utrecht Medical Center. So what you're looking at on the left is a bright field picture of a transverse section of an explanted ACM heart, by which you can see in yellow the fibrofatty deposits that clearly are surrounding this myocardium. On the right, you're looking at a meson trichrome of the same heart, and you can see that the muscular content of this heart is severely compromised, with a lot of the fibrofatty replacement taking over a large part of the myocardium. So what drives this phenotype in this particular patient is a mutation in plakophilin 2, so one of the desmosomal proteins. It introduces a premature stop, which causes haploinsufficiency in these patients, so they simply have less of the PKP2 protein. So to get a feel for the gene expression changes that are occurring in these ACM patients, what Kees did, he made use of an approach which we call tomo sequencing. So tomo stems from the Greek word tomos, which basically means section. So it's basically section sequencing. So what Kees did, he took a rim of tissue spanning from the epicardium more towards the endocardial region of this explanted heart. He cut it in 220 different sections, barcoded each section, and then sequenced these sections. And what you can then do is link local remodeling responses to a certain gene expression profile. And in doing so, when he looked at the gene expression profiles in these individual sections, going from the epicardium to the endocardium, he could clearly see a separation and basically clustering the sections more towards the epicardial region, and also clustering the sections that were basically covering the endocardial part. What tomo sequencing allows you to do is look at the expression pattern of several marker genes. So for example, PLIN1 and FAP-4 representing adipocyte marker genes, you can see that they were more abundantly detected in the epicardial region, which is of course expected if you see that that's the fibrofatty region in this particular patient. You can also look for markers for fibroblasts and cardiomyocytes. And so you can locate the different cell types using the expression pattern of these particular marker genes. So what this also allows you to do is look at genes that are specifically enriched in the regions that are being actively remodeled. So what we are hypothesizing is that the regions in which the cardiomyocytes are degenerating and where the fibrofatty cells are taking over, that's actually where all the activity still takes place. So if we can identify the mechanism that causes these cardiomyocytes to degenerate, we might actually be able to interfere with this process and thereby maintaining a better integrity of the cardiac muscle. So one of the genes that we found specifically induced at these sites of active remodeling was ZBTB11. This is an ill-studied zinc finger transcription factor, but when we then looked at a staining pattern, so the immunocytochemistry or histochemistry of ZBTB11, we could clearly see that ZBTB11 is induced in the cardiomyocytes flanking these fibrofatty patches. This is a higher magnification of this, as you can clearly see that ZBTB11 is more highly expressed in these specific cardiomyocytes. So this was not just due in this specific patients that we tomo sequenced, but also looking at the ZBTB11 expression in additional patients suffering from genetic cardiomyopathy, also having not only a PKP2 mutation, but also mutations in either desmin or CRYAP, we could see that ZBTB11 was induced in a specific subset of cardiomyocytes flanking these fibrofatty regions. So next case wanted to see what the function was of ZBTB11. And to study this, he made an adenovirus, adeno-associated virus, overexpressing ZBTB11, and he next infected cardiomyocytes that came from iPSCs. So in doing so, he was able to overexpress ZBTB11 in these human cardiomyocytes. And what he next saw is, after a whole set of in vitro studies, that ZBTB11 overexpression in these cardiomyocytes indeed resulted in an increase in apoptosis. And at a gene expression level, you could see that there was a strong induction in genes related to p53 signaling. So really pro-apoptotic signaling was induced in these cells that were exposed to a higher level of ZBTB11. So what we now think, and we have many more data, is that because by using tomo sequencing, you can really identify the regions that are being actively remodeled. You can identify genes that are induced in these regions and likely play a role there. And then by functional follow-up studies, we could actually show that ZBTB11 is an active player in cardiomyocyte degeneration. And this is something we're now following up on. You can also take it a step deeper, of course, and look at the single cell level. As with tomo sequencing, you're still sequencing a couple of thousand cells per section. So what we did in our lab, and this was an effort driven by Monica Gladka, she really optimized a protocol that we can use to perform single cell sequencing on all the main cardiac cell types from an adult heart. And this is of course tricky, as we all know how fragile and large our cardiomyocytes are. But what we came up with was a protocol by which we took the tissue. You need fresh tissue, you digest the tissue, you put it through a flow sorter, and then by using a large nozzle, you can basically collect all the single cells in a 384-well format. We performed this on a mouse adult heart, and that worked out really nicely. So we next set out to do this on human disease tissue. And of course, because we need fresh tissue, this really made our options limited. But there's one genetic cardiomyopathy that actually grants us access to tissue, which is hypertrophic cardiomyopathy. So often these patients have a mutation in one of the sarcomeric genes, like for example, myosin binding protein C3 is a key gene that's often mutated in these patients. And these patients suffer from septal thickening. So both of their walls thicken, but it's mainly septal thickening that is causing problems by causing an outflow tract obstruction. So what the doctors do to these patients is they basically get a septal myectomy. So they basically resect a part of your septum so the obstruction is removed. So we could take this tissue, disperse it into a single cell suspension, put it through the fax, and then collect individual cells. So this is of course a very unique opportunity to get access to diseased cells. And we were very happy to see that it worked out. So we first started off by looking at whether our cells were truly diseased. So what we did is, of course, we have had difficulty collecting healthy cells ourselves. So we made use of available data sets and looked at key marker genes of disease. So genes like MPPA, B, ACTA1, MYH7, also known as beta-myosin heavy chain. And we could clearly see that compared to the two control cardiomyocyte sets, these stress marker genes were more highly expressed in our cells. So indicating that our cardiomyocytes that we collected were more diseased than the control cells that were previously analyzed. Because cardiomyocyte heterogeneity is a key issue in hypertrophic cardiomyopathy, we next focused in on all the cardiomyocytes that we collected in our study and performed clustering analysis. And in doing so, we were able to identify 6 different clusters of cardiomyocytes, to which all of the 5 patients from which we collected cells contributed to. And you can see that, for example, there's a cluster that's more enriched for MPPA, a cluster that's more enriched for XERP2. So it's really the different cardiomyocyte populations in these particular HCM patients. However, when we looked at the clustering analysis and compared this, whether this was also happening in the control cells, we could clearly see that cluster 1 to 4 were more strongly present in the HCM cells than in the control cells, meaning that the clustering of these genes is more predominant in HCM cells. So really showing that the clustering that we're obtaining is HCM related. As I mentioned, MPPA was one of the genes that showed an enrichment in one of the clusters. So looking at MPPA, and not surprisingly, we could detect that MPPA is more enriched in cardiomyocytes coming from our HCM patients. Also by staining, we could confirm the presence of MPPA positive cells in the HCM explant, while it's absent in the control heart. And the localization actually appeared to be close to the fibrotic areas in these patients. So obviously one key benefit of single cell sequencing is that because you have the gene expression profile in this particular cell, you can do a correlation analysis of genes. So what we did is we looked for the genes that show a strong correlation to MPPB, either a positive or a negative one, MPPA. Of course, we detected MPPB because MPPA and MPPB are often co-expressed, but there were also some surprising candidates among the positively correlated genes to MPPA. So it really allows you to identify new genes that might be playing a role in these stressed cardiomyocytes, as we know that MPPA is a marker for cardiomyocyte stress. Surprisingly, however, we saw a negative correlation to genes like titin, the ryanodin receptor, and NRAP. So this is also something we're currently following up on. When we looked at the expression level of the particular genes correlated to MPPA, we could see that they showed a higher expression in the cardiomyocytes coming from these HCM myectomy samples. Next to gene correlation, you can also look at panels of genes, whether they show a similar correlation. So, and this is what we call gene modules. So it's really sets of genes that appear to be co-regulated in all the different patients that we tested. So in doing this bioinformatic analysis, Martijn Behrens in our group, he came up with 5 different gene expression modules, as you can see here, with one, for example, being led by MIL2. Oh, I'm sorry. So also these gene expression modules, they show a different expression pattern across all the cardiomyocytes that we can identify. And of course, this is only an NS5 that we performed single cell sequencing on, but luckily we also had access to roughly 100 additional myectomy samples in close collaboration with Jolanda van der Velde and Michelle Michels. And also in these samples, we were able to look at and validate the gene correlations that we found to be happening in the 5 patients we performed single cell sequencing on. As we are using flow cytometry for our single cell sequencing, to collect our single cell sequencing data, you can also index sort your cells, meaning that you can link cell morphological parameters to the gene expression profile in an individual cell. As cardiomyocyte hypertrophy is, of course, a key aspect in hypertrophic cardiomyopathy, we can now look at the gene expression profile that's expressed in the larger cells. And this is just something we only performed in 2 patients so far, but you can clearly see that the genes coming from module 2 are expressed in the larger cells in these 2 particular patients. So we're very curious to see whether this is also happening in more patients. But then when we do perform staining and correlate this to cell size ex vivo, we can also see that MIL2 is higher expressed in the larger cardiomyocytes. And of course, this is very intriguing and interesting to follow up on. And of course, you can look at additional cell morphological parameters also. Something that I'm very excited about myself too is the iPS-derived cultures, of course, especially related to genetic cardiomyopathy, because you can take your iPS culture harboring your pathogenic mutation, basically differentiate them in all the cell types that you're interested in, and then study the disease phenotype and compare it to its isogenic control. So In our lab, Brian Sebastian van Kampen, he's actually the go-to guy if you want to gene edit anything. So he also took an IPS line containing a PKP2 mutation, the C2013-del-C. He reverted it back to wild type. So we had an isogenic control for this IPS line. And then Arwa Kohela, a PhD in our lab, she was very intrigued by the fact that this fibrofatty replacement appeared to have an epicardial origin. So what Arwa did is she took these IPS cells, both the mutant ones and the reverted ones, and she set out to optimize a protocol to differentiate these cells in epicardial cells. And she was able to do so based on this marker gene expression, WT1, which is a key marker gene for epicardial cells. She could see that both the mutant and the reverted IPS-derived epicardial cells were positive for WT1. Up until day 28, the WT1 expression was comparable in both the mutant and the reverted cells, and this she could also quantify. So while they were as epicardial-like still after a month of culturing, when we performed RNA sequencing on these cells, we could clearly see that already in the mutant cells were very different than wild-type epicardial cells. And then when we looked at the functional annotation of the regulated genes, it was very striking to see that there was all kinds of functional annotations like dilated cardiomyopathy, cell adhesion, extracellular matrix. So really aspects that are really related to ACM already. So it's really a gene expression difference in these epicardial cells that already are related to the disease itself. So one of the functional annotations that we saw was adipogenesis. And this is, of course, very interesting if you think that there's an epicardial origin, potential epicardial origin to the fibrofatty replacement that we are seeing in this patient population. So when we looked at marker genes for adipocytes or fibroblasts like PPAR gamma and fibronectin, we could see that the mutant cultures compared to the reverted cultures indeed were expressing higher levels of adipocyte or fibrogenic markers. And when we also looked at an oil red O staining, so really looking at fat droplets in these mutant cells compared to the reverted cells, we could see that in the mutant cells there were fat droplets present. So really showing that the mutant epicardial cells by themselves spontaneously become more fatty-like. So we performed single cell sequencing. We looked at the genes that were specifically expressed in the mutant cells, so the magenta cells compared to the pink cells, the healthy cells, and we looked for transcription factors that were more strongly induced in the mutant cells. And by a bunch of bioinformatic and in vitro experiments, we identified a transcription factor known as AP2 or TFAP2A to be induced in the mutant epicardial cells. And also when we look at explanted heart tissue from ACM patients, we can see an induction in TFAP2A in these epicardial layers. At the same time, Brian wanted to see whether we could make a humanized mouse model of this particular mutation. So what he did is he basically knocked in the comparable mutation in the mouse genome, which turned out to be the 755-del-A mutation, giving the exact same amino acid change as was happening in the patients. So also here, we were happy to see Jenny Chu from our lab. She performed all the mouse studies. She was able to show indeed a haploinsufficiency for PKP2, both at the protein and at the RNA level, which we could confirm. But not only did we see a decline in PKP2, but surprisingly, in response to the single point mutation in PKP2, we saw a complete desmosomal degradation. And this is very striking, especially if you look at the fact that the floxed allele of PKP2 does not show this phenotype. So there's something specific about this point mutation that's pathogenic compared to the floxed allele that has been previously used. We could see a desmosomal degradation, a decline in the protein level, which was not happening at the mRNA level. these animals started showing signs of cardiac dysfunction and remodeling. And when we performed proteomics analysis, in addition to a decline in all these desmosomal proteins, we also saw a decline in cadherins and adherens junctions proteins, and an upregulated in these proteins related to protein degradation. So this is what we're now currently following up on in detail, because we're of course very intrigued to see what drives this proteasomal or this desmosomal degradation. And even more interesting to see is that this is not just a mouse phenomenon, but this is actually also what we're seeing in patients that have a PKP2 mutation. So all these 4 ACM patients have a different mutation in PKP2. And also here, we're not seeing only a decline in PKP2, but it really seems to result in this desmosomal degradation in general. So we now think that if you can rescue PKP2 degradation, you can actually have a therapeutic benefit. And this is what we're also seeing in our initial experiments there. So I want to stop right there. I hope I have shown you that we think that you can use tomo sequencing if you want to look at the local remodeling responses and really want to get a feel for the gene expression differences that are driving this heterogeneity in remodeling across a certain tissue. If you want to go for single cell analysis, it really allows you to look at the differences between the different cell types, but also within one cell population at cellular heterogeneity, which we, especially for HCM, think is very relevant as these cells respond so differently. I think IPS-derived cultures are giving us a wealth of information, new information that will help us to understand these genetic cardiomyopathies better. With CRISPR-Cas, you can make very relevant disease models, I think. And of course, gene editing is still in its baby shoes, but I have no doubt that this will be a big focus for many of us in the near future. So I think I mentioned most of the people involved along the way, but this is the team and the funding agencies and the collaborators. And I thank you for your attention.
Thank you very much, Eva. And we will have a discussion at the end of the webinar. And let me pass the chair to my co-chair, Professor Christian Weber. Christian.
Yeah, thank you, Tom. It's my pleasure now to introduce Sebastian Cremer, who provides the second part of our lectures here. Sebastian is a clinician scientist and a cardiologist, actually from the Department of Cardiology in Frankfurt, where he trained with Andreas Zeyer. And he also did some seminal work on the functions of long coding RNAs like MALAT, for instance, in atherosclerosis, where we also had the pleasure to collaborate in Stephanie Dimler's lab. And he then went on to do a stay in another powerhouse of cardiovascular research in the Nahendorf lab, and we are looking forward maybe to him sharing some more exciting insights from his new research. Sebastian.
Yeah, thank you very much, Christian. I will talk about hematopoiesis and cardiac inflammation in recurrent myocardial infarction. So this is not directly linked to, this is not directly a cardiac remodeling study, but inflammation after MI is clearly linked to remodeling. It is known for a long time that myocardial infarction elicits an inflammatory response where leukocytes, which are produced in the bone marrow, invade the infarcted tissue and the surrounding myocardium, and inflammation peaks around day 3 or 5. And it is assumed that in the first wave, so in the first days, these invading monocytes, so the invading leukocytes are mostly monocytes, are rather inflammatory, and on the long run, they contribute to restoring cardiac function. So this is what I mentioned. The bone marrow is very central in supplying those leukocytes to the myocardium. And very recently, a concept which is called trained immunity was introduced, which says that preceding inflammatory stimuli or challenges shape myeloid cells, monocytes, but also HSPCs, which overreact to a second inflammatory stimulus. For instance, beta-glucan is such a stimulus, but also factors which contribute to development of atherosclerosis, like Western diet, promote trained immunity. So we hypothesized that inflammatory signals which induce emergency hematopoiesis after myocardial infarction would have consequences for inflammation in recurrent cardiovascular events. Why is this important? So in a relevant proportion of patients who have ST elevation myocardial infarction, so an occluded coronary vessel, recurrent MI occurs, and this happens very shortly, usually after the index infarct, so within a week or at least within 1 month. And this is for those patients very important, because patients who suffer from a second MI or recurrent MI have a really worse prognosis compared to patients who only have one myocardial infarction. And I mean, the assumption is that surplus of necrotic myocardium contributes to this or is responsible for this adverse outcome, but inflammation in this context has never been studied. To do this, we developed a mouse model where we can separately ligate the circumflex coronary artery in mice from the LAD, so in 2 different procedures. You can see this here. This is fluorescent angiography, where the different points of ligations are highlighted, and the LCX ligation, or the circumflex ligation, will create a lateral infarct, which is smaller compared to the LAD ligation, and combined we have a very huge myocardial infarction. This is also illustrated here in these MRIs of mice. This is the first infarct, the circumflex infarct, and this is when we combine both infarcts, which creates a very huge infarcted myocardium. We started with the circumflex infarct because it's very difficult to do a second surgery on a mouse. And for the lateral infarct, we have to lift up the heart a little bit. So we evaluated this in MRI also. The LCX infarction, or the circumflex infarction, will only slightly reduce ejection fraction. But if we combine both, we have severely reduced ejection fraction and also adverse remodeling with increasing end diastolic volumes. Of note, after a myocardial infarction, after 10 days, the bone marrow activity, which is central for the following understanding, returns back to normal. So leukocyte numbers, which increased after a myocardial infarction, or also HSPC numbers, which increase in the marrow, return back to baseline after 10 days. So this is the results then. In gray, there's a mouse which only received an LID ligation, and in red, the plots show mice which received or experienced a circumflex ligation previously. We chose a 10-day time span because bone marrow activity returned back to normal after 10 days, and also because 1 week or 10 days is a relevant time frame for trained immunity. Surprisingly, we saw reduced inflammation in mice who received a second infarct compared to mice which received an index MI only. So there's less monocytes in the peripheral blood, and there's also a tendency towards less neutrophils. And this is also reflected in the bone marrow. So we have less HSPCs and specifically less GMPs, and also less proliferation of HSPCs. And most importantly, we observed reduced infiltration of leukocytes into the infarcted myocardium when mice experienced an MI previously. Because we were interested in the bone marrow, we checked for niche factors in the marrow, and what we could observe here, that in mice who received or who experienced 2 MIs, quiescence factors in the marrow like KIT ligand, VCAM1, or also CXCL12 were higher compared to mice who experienced the first infarct. We tested the same thing in transient ischemia, where we did ischemia reperfusion for 30 minutes because this is more relevant in the clinic. So troponin release was similar in both groups. And again, we observed a reduced proliferation of HSPCs after transient ischemia, or in mice who received an MI after transient ischemia previously. And also we observed here less infiltration of inflammatory leukocytes in the infarcted myocardium. To test whether this is a local event or also whether this is caused by, for instance, innervation of the sympathetic nervous system, or whether this is a circulating factor, we did parabiosis experiments where one parabiont, which is labeled in red, received MMI, whereas the other did not. And after splitting the mice, the other mouse received a myocardial infarction, whereas in gray, the control mice didn't receive surgery prior to separation of parabiosis. And what we observed here, again, is reduced HSPC numbers in the marrow of mice where the partner mouse received an MI previously, and also reduced proliferation of HSPCs. And this is also reflected in circulating leukocyte counts, and also we observed less infiltration of inflammatory cells into the heart. Again, we tested this also after another with another stimulus, and so not an MI, so mice who received an MI and were challenged with LPS. Again, we had reduced HSPCs in the marrow and also reduced proliferation. Because myeloid cells, and especially monocytes and macrophages, are very important for trained immunity, and because also macrophages are an important part of the bone marrow niche, we tested the hypothesis that macrophages would mediate these effects. And for this, we sorted, or we isolated macrophages from the bone marrow. You can see the gating strategy here. So macrophages in the marrow express MHC class II molecules and are positive for CD169 and VCAM1, but do not express CX3CR1. And we also isolated other niche cells like endothelial cells or mesenchymal stromal cells, and assessed niche factors in those cell fractions. And what you can see here, that after an index MI, which is labeled in blue, in macrophages, which is panel E on the very bottom on the right, maintenance factors like VCAM1 or angiopoietin are reduced after a first MI, and this is not the case after a second MI. So we think that this tolerance phenotype we see could be at least in part mediated by macrophages. We tested whether this whole framework is also relevant in patients after myocardial infarction, and retrospectively analyzed leukocyte counts in patients who experienced 2 MIs compared to 1 MI. You can see that the infarcts were, with respect to troponin release, fairly similar. You can also see that a first MI resulted in an increase in white blood counts in patients, whereas a second MI, which is labeled in red here again, did not yield increased white blood cells in the circulation. The increase which was there is very reduced compared to patients who received an index MI. In addition, another group tested the same thing after, in patients who had stroke recurrence, which is maybe even more important, and even also here, leukocyte counts were reduced after a second stroke. So to sum up, we could demonstrate that the first infarct, which elicits a very strong immune response, shapes the immune response to another recurrent infarct, where we have a mutant inflammatory response. We assume that the bone marrow is implicated in this phenotype, and especially macrophages who have different expression of hematopoietic maintenance factors. And why is this important? It has been known for a long time that insufficient inflammation after myocardial infarction is associated with a worse prognosis. So this is a large analysis of more than 10,000 patients here, and you can see that patients who have very little leukocyte counts on the left side experience higher mortality. And also, anti-inflammatory drugs enter the clinic, at least for atherosclerosis, but also maybe for MI patients. These are data from the COLCOT trial, which show a beneficial effect of, for instance, colchicine after myocardial infarction. And these drugs should maybe utilized with caution in patients with recurrent MI. And with this, at the end, and I'd like to thank everybody who was involved in the study, especially Matthias, who was my supervisor in Boston, and also Maximilian Schloss, who helped me a lot with the surgeries, and I'm happy to take your questions.
Yeah, thank you very much, Sebastian. And so we join a shared discussion for both presentations now, and I encourage you to post your questions in the chat and the questions and answer tool here. So maybe, Tom, do you want me to start a little bit?
Yes, if you can start, Christian, that would be great.
I would just come to the last presentation, because that was instigating quite some interest. So it was well known and also described by the Nahrendorf group that acute myocardial infarction also exacerbates atherosclerosis. So the question then arises whether this phenotype that you observe on recurrent myocardial infarction would also induce some tolerance as to the effects that exacerbate atherosclerosis after a first MI.
Yeah, so I think the main difference between those two studies is that we induced the second infarct very, very close to the index MI. So this was 10 days afterwards, whereas the atherosclerosis study Pata did, I think in 2012 or so, had a chronic disease with a chronic phenotype. And also in this study, the sympathetic nervous system was responsible for the phenotype, whereas we have a circulating factor here. So maybe there's a difference between an acute and a chronic response, but it's clearly, yeah, it clearly differs.
Okay. And related to that, what was the difference between the events in humans? And was there this sort of lack of response of leukocytes that is quite interesting in the second MI, dependent on the length time between the infarcts?
So these were not too many patients, I think around about 40 patients. You know, in clinical practice, a second infarct usually occurs very soonish after the index event, either because patients do not take their platelet inhibitors, or maybe because the stent wasn't implanted so well. So, but what we usually see is this within a week or so. We very rarely have a second coronary occlusion, let's say, after 2 months or even longer. And this is also what the data of the Horizon AMI trial showed.
And there's also one question here in the chat. What about neutrophils? Did you look at that, and would that also be altered in a recurrent MI?
So we also looked in neutrophil infiltration into the heart, and also neutrophil levels in blood, and it was like it went into the same direction like monocytes.
Okay.
Emma Robinson was asking Eva about the tomocyte data and the potential for identification of sort of pro-inflammatory role of the fibrofatty deposits contributing to this local remodeling. Could you comment?
Yes, of course. Very good question from Emma, because we also think that inflammation plays a key role in the remodeling process in these patients. I don't think tomosequencing is the ideal way to study this. So what we're doing now is looking at iPS-derived monocytes and cardiomyocytes to see whether there's a crosstalk between the two based on receptor ligand pairing. Because we do see that there's an increase in cytokine secretion in these patients, pro-inflammatory.
I can see AJ has a question.
Eva, yeah, it's always a delight to listen to you. Really a pleasure. So The degradation of desmosome proteins is quite intriguing and has been observed repetitively by independent investigators. We first reported that in 2014, and many others have seen the same thing. But none of us have figured out the mechanism. I know that Farah from UCSD has reported COP9 being a factor which we don't see in our models. What is your explanation when you mutate plakophilin 2, other desmosomal proteins get degraded?
Yeah, so we also don't see a change in the COP proteins. So I don't know why that is, but maybe that's because she's looking at a DSP related. I'm not sure.
No, I mean, a fake model. She deleted COP9, homozygously said the proteins get degraded. That's not what's happening in the RBC. That's not a deletion of it. So the model is a fake model in that regard.
I'm going to let you fight that battle.
What do you think is the
underlying? So what we're thinking is that PKP2 is really a key anchoring protein for the desmosome. And as soon as you have lower levels of PKP2, all these desmosomes have basically no place to home anymore. And then they start floating. And as they start floating, they become degraded by UPS. So what we've seen is that if we have a UPS inhibitor, we can actually block this desmosomal degradation. And what we also know is that if you overexpress wild-type levels PKP2, you can restore the phenotype. So we really think that
PKP2 simply failure to localize to the proper place. Is that
that's what we think.
Very good, thank you. The other thing that you described two transcription factors, but you didn't elaborate on their function. One was ZBTB11, and what does it do in the heart, which is a diffusely expressed transcription factor with a lot of other functions with questionable relevance to the heart in terms of craniofacial abnormalities that it cause. The second one was your transcription factor science translational research, that or science translational medicine, whatever the name of that journal is, TFAP, and how does it regulate adipogenesis?
Yeah. So from TFAP, we know that it directly regulates these adipogenic genes. So we've done some Homer analysis, and we basically know the target genes for TFAP2 in these particular epicardial cells. CBTB11 is a lot less clear, so it is very broadly expressed indeed. The only thing we know is that it's induced in these cardiomyocytes that are flanking these fibrofatty regions. So I think it needs an additional signal because we don't see it increased in ACM-derived cardiomyocytes. So we don't know a lot there.
Like all good discoveries, there are more questions. Thank you.
We have a methodological question from one of the participants about ensuring that your cardiac myocytes are alive and intact.
Yeah, that's of course a key question as we are using flow sorting. So that's actually also one of the comments we have been getting quite a bit. So we put in a lot of efforts to make sure that the cells that we are collecting are single cells, viable cells, and that they are also intact. And to do so, we are gating them for size, autofluorescence, DAPI, and DRAG5, which is actually only in intact nuclei. So we are gating in different directions to make sure that we are getting the live cardiomyocytes. But good question.
Another question following up on that, by Magdi Elabal, she noted that the cells you were analyzing were mostly of left ventricular origin, and she's wondering actually whether you had any samples also from the right ventricle, and whether the gene expression profile actually is resembling that in dysplastic right ventricles.
So that's related to the single cell analysis?
Yeah.
Yeah, so no, we're restricted to these septal myectomies. So we can only get cardiomyocytes from the septal region in these patients, because we need fresh tissue for our approach. If you go for a nuclear sequencing, you can obviously use frozen tissue, which would broaden the applicability.
Okay, then maybe another question, getting back to the also maybe single cell profiling. There's a question to Sebastian by Sumaya Benaki, whether you also look at the immune profile of macrophages in the myocardium, so in the infarcted area, not only at your quiescent phenotype in the bone marrow, and whether that basically changed in recurrent MI.
Yeah, this is a good question. So we didn't look into this. So we just measured monocytes and macrophages and didn't profile them further. But this, if we would like to follow up on this, that would be a great idea, sure.
And probably another related question is about immunoregulatory populations that was asked. So did you look at that or not?
Yeah, as I said, we didn't phenotype the leukocytes in the myocardium further in this project, but it's yeah, it's a clear possibility that this also alters recruitment of new cells, but we didn't follow up on this.
I would also have a more principal question. I mean, myocardial infarction is one very prototypic type of ischemic injury, but there are also other ischemic injuries. So do they share this phenotype, or is this like, for instance, if you would have just like an ischemic muscle injury, or I think there's also some people studying the effect of stroke on repetitive injury and also atherosclerosis. So is this a specific signal from the myocardium, or is this also shared in this phenotype and other types of ischemic injuries?
Yeah, so as you mentioned, recurrent stroke is from a clinical perspective, maybe a larger problem. And there are also people studying the effect, let's say, epigenetic modifications or changes in leukocytes or in brain leukocytes after stroke, and I know preliminary data which might show similar reactions. Also, patient data after stroke show reduced leukocyte counts after recurrent stroke compared to an index stroke. So I don't think this is restricted to a myocardial infarction, but also to other ischemic injuries which activate the marrow. So this is my assumption, at least. I cannot prove it. But
if I remember well, in stroke, but I may be wrong, the lymphopenia is kind of a strong predictive factor. Is that a fact? And would that then your observations could somehow be linked to that? I wonder.
Yeah, so I'm not really sure about lymphopenia and outcomes after stroke. I would rather assume that lymphopenia is not good for anything in any disease. But yeah, maybe so. My idea is that there needs to be a very good balance in when it comes to hyperinflammation, but also too little inflammation, and both is not good.
And then there is another methodological question to Eva regarding the proteomic approach used in single cell. What kind of methodology, instrument, and so on did you use, if you can comment?
I would have to refer this person to Manuel Meyer. We collaborated with him on this aspect. It was indeed through mass spectrometry, but I don't know the details regarding the machine.
So they can, Aline can email Dr. Meyer. Very good. Any other questions or comments? Anna Rita is asking here. I haven't read it yet. 1 second. Christian, do you have any?
Yeah, I'm also trying to collect this next question, where it's basically about one of the genes dramatically upregulated in HCM, RTN4, which gives rise to NOBO-A, obviously, in the heart and the R membrane protein regulating serum metabolism. And ceramide has also been implicated in major cardiovascular events and also cardiomyocyte autophagy. And so basically, she's asking whether you would propose to look further into this, for instance, by correlating RTN4 expression and ceramide profile with the pathology in your sample. So this is a very specific question, but an interesting one.
Yeah, no, so we haven't looked into the function of RTN4 yet, but we do know about its proposed function, which might be indeed relevant. So we'd like to follow up on it. And then there was also somebody asking about the protocol of the single cell sequencing on the cardiomyocytes. That was published by Gladka et al. In Circulation a couple of years ago, 2018. But if there's any specific questions, you can always email me and we'll help you through it.
That's great. And I think a final question, or maybe final, will be asked by AJ, but let's ask the second question from Emma.
So Eva, what is your current thinking about the signal that leads to cell death in arrhythmogenic cardiomyopathy? What is the underpinning stimulus that cell senses in order to go either apoptosis or necrosis or other mechanism of death?
Yeah, I think it's mitochondrial dysfunction. So I think it's energy production within the cell. That's a problem. What do you think?
How do you link that to desmosomes?
Yeah, that we don't know, but we do see that as soon as you have well, we know this based on our mutant cells, and if we dose in an SI against PKP2, it one-on-one correlates with a decline in mitochondrial function.
That would be an interesting. Thank you. You look into that.
And maybe let's end with a question from Emma, although some more questions appeared, but Emma Robinson asked about the properties of the fibrofatty sort of deposits that you are observing. Whether what is the thermogenic gene expression? Are they sort of gray? Are they brown, or what kind of a color they are?
Emma, you would have to tell me. I don't know. So I don't know the details there. So, but an interesting thing to look at. And then related to the homozygous knock-in, mouse model, the PKP2, they don't live. So non-viable.
Wonderful. So we have essentially covered all questions, and perhaps, like Eva said, if you have any additional questions also to Sebastian, maybe just contact them by email. I think this is not the end of interaction here.
We strongly encourage. Christian, would you like to summarize?
Yeah, I think we had another round of two very interesting presentations, very complementary this time, and covering really a large area of cardiac and cardiovascular biology. And I hope that we can continue to enjoy this format also in the new year. This is basically our last webinar this year, and so Zara will be in touch with everyone and basically invite you for the new series in the next year. And it has been a pleasure the whole year actually to do this with Tom together, and also contributing to the visibility of cardiovascular research and also to the Basic Science Council.
Thank you very much.
They're very popular seminars. They're very well attended. They're very popular, and we have great speakers. Thank you all.
Wonderful. So Tom, do you want to conclude, or?
Yeah, well, yeah, on behalf of both of the organizers, both the journal and the Basic Science Council of the ESC, thank you very much for joining us here today. Maybe it's also a good opportunity, Christian, to emphasize that next year there will be a Congress of Basic Science organized by the ESC that you should all look out for, the Frontiers in Biomedicine. Please check this out and submit and consider coming to the conference to Budapest. Yes.
And it will be physical.
And it will be physical.
Okay.
Thank you.
April 29 to May 1st in Budapest.
Thank you very much.
Thank you very much.